Good afternoon, everyone. Yes, this is my first presentation in this platform. So today I am going to tell a journey towards the establishment of a molecular laboratory in the remote area in Bihar, as well as the glimpse of the molecular findings in the same lab. So today I represent Homi Bhabha Cancer Hospital Research Center, Muzaffarpur. So this is the center under Tata Memorial Center in Muzaffarpur district, Bihar. It has been established in early 2021. The aim was to provide quality cancer care accessible and affordable in the rural areas of Bihar through its state-of-the-art diagnostic as well as treatment facilities. So we know nowadays that genomic profiling and the use of targeted therapies have now become a part of the cancer management. So keeping this in mind, one molecular laboratory was conceptualized to be established in this hospital. So this is the timeline. In early phase of 2022, the planning was initiated how the project will be established, followed by the funding, infrastructure setup, and procurement of the equipment. In mid-July, in the mid of 2022, the lab was functional. Then lab manpower hiring, followed by their proper training, then panel development, validation, method standardization. The lab started the providing the service to the patient at the end of 2022, and the service was basically the next generation sequencing to identify the alteration at the genome at the cancer patients. Entire 2023, the lab services were continued along with facing multiple challenges, overcoming those challenges, and at the end of the year we were able to include one more test, that is qualitative RT-PCR based single gene mutation detection. And now in this year we are smoothly running this lab with the sequencing as well as these RT-PCR based tests. So till date, starting from December 2022 till June, we have total number of 393 samples processed at our lab. Out of them, 330 samples were for the NGS and 63 samples were for the real-time PCR. And this is the Bio-Rad-based platform with the commercially available kits from the Diatech Pharmacogenetics. Let's have a more on our NGS platform as well as the findings. So ours is a mini-seq platform. We are using one 72 gene panel with 67 genes that can easily identify these three alterations, SNP, index, copy numbers, alterations, and 11 genes for the fusions, and this is hybrid capture based method. So starting from the sample 1 to till date, we are actually maintaining good QC matrices. Some of the instances are mentioned here. At the pre-analytical phase, if the tumor content is less than 20%, if the tumor size is very tiny, those samples are rejected outright. If samples are processed for extraction, post-extraction we do the intactness of the DNA, that is actually identified by this DNA integrity number. We maintain one proper threshold here. If this is below that threshold, we reject those samples. After the library preparation, even we measure the peak of the library, and there also, if the quality is below the threshold peak size, those samples are not included in the sequencing. During the sequencing, Fred score, that is the Q30 score, is maintained more than 85% as per the mini-seq specification. Depth of coverage is maintained 1000x. If the mean depth is less than 250x, those sequenced samples are not taken further for the processing. So now this is the compiled data of total solid tumor cases for those NGS were performed. Here you can see 68%, a big chunk of cancer is from the lung, followed by gallbladder, then GI, that includes colon, rectum, esophagus, sarcoma, bladder, prostate, breast, ovary, head and neck, that includes adenoid cystic carcinoma, anaplastic thyroid carcinoma, Then this is the molecular interpretation of all the cases we have performed here. You can see again the big chunk is the lung cancer, and these are the other cancer types. Lung cancer, here I just present the non-small cell and the small cell lung cancer. And for both the subtypes, those actionable driver mutations are actually here mentioned in this black colored, and their relative abundance is represented by this size of each of the genes. This is altogether lung profile, and here you can see the other cancers. Here even some of the actionable mutations are very prominent, like EGFR, exon 19 deletion in esophagus, KRAS in rectum, KIT in GIST, BRCA, PIK3CA in breast and ovary. So let's have a more detailed interpretation of the lung cancer cases only. So I here present only the first year lung cancer cases, which is only maybe the number is very less, 70 cases. Here even you can see 67% is of the adenocarcinoma, and other subtypes like small cell, squamous, large cell neuroendocrine, sarcomatoid, germ cell tumor are also present, but less in number. 40 to 60 and more than 60, this category has the maximum load of patients here. And here we can see that baseline cases are 85.7%. Molecular interpretation of these lung cancer, you can see, EGFR mutation harboring cases is the highest, that is 39%. Except EGFR, other actionable mutations of genes like comprises of 36% of cases. That includes ALK fusion mutation, ROS1, ERBB2, MET, RET, BRAF, KRAS. And apart from these actionable driver, other driver genes are present in 25% of the cases. So since EGFR mutation harboring cases is the highest, so little bit we are now zooming in those cases only. Here you can see again the female represents the highest in number, and this here it is the smoking pattern. This see the middle panel, this non-smoker, which is 67%, and here female 41%. So again the non-smoker is a highest chunk among these smoker, as well as data not available these cases. And those four most predominant exons of the EGFR, here you can see exon 19 deletion is the highest one, 67% of the cases, followed by exon 21 L858R, and then comes the exon 20, that is mainly insertion deletion, and exon 18 is the lowest one. Those EGFR harboring cases also shows the mutations in the other genes as well. Here are those other genes I can just show here in this table. And interestingly, again, P53 occupies the highest one with 34% of the cases, followed by the CDK N2A in 11% of the cases. So some of the facts actually corroborates the already published literature, like adenocarcinoma is the highest in the lung cancer, female non-smoker is the highest in this EGFR harboring cases, EGFR mutation is the highest in out of all the alterations, TP53 comes in the highest altered gene co-occurring with the EGFR, and fifth point is that P53, here we can see that exons 5 to 7 are primarily altered in our cases, and the published literature also shows that exon 4 to 8 actually harbors the DNA binding domain, and any kind of alteration in this domain actually affects the target gene binding of P53. So this is all about our NGS finding. So just very briefly presenting our data of single gene mutation detection. Here we perform the RT-PCR based mutation of the EGFR gene, 3 isoforms of RAS, RAF, and polymorphism of DPYD gene as well. So, so this is all about our molecular data. Now you can see that this high-end molecular lab is running despite this is our functional unit of this hospital, current status. This is the administration OPD area, and actual our main building construction is undergoing. So running such a high-end lab in the premises of one state-run medical college was a biggest challenge in our scenario, because this machine, this lab requires continuous maintenance of the AC, temperature, humidity, voltage, earthing, so many things, actually uninterrupted power service, everything we actually needed to sort out during the run of this entire sequencing process, convincing those management, make them sensitize to get it done within the time. That affected actually our turnaround time in between. But yes, entire troubles were actually, we were able to overcome all the hurdles because of the support of my seniors. We are fortunate to have them. First comes Dr. Ravikant, who is the in charge of this hospital and super positive of this laboratory. This is my small team, consists of these young GRFs. Actually, this is also was a very big challenge, getting the appropriate manpower coming from the appropriate academic background with the correct attitude. That was very tough initial days. And even another point, actually, I must mention that even getting the nucleus-free water, getting the mol biograde ethanol, getting the appropriate make plastic ware, that were very tough initial date, and all those things actually affected initially our smooth run of this service. Anyway, so frequent visit of Kumar sir, frequent visit of Pankaj sir actually sorted many of the technical difficulties. Amit sir, because of whom one research atmosphere was established in the lab. Anuradha ma'am and Pratik, who are associated with this lab from day one. We have learned many things from ma'am, starting from the lab management, sample management, extraction procedure, how to overcome the challenges. So this is all because of ma'am and Pratik. Apart from that, Doctors for You, who actually supported in the establishment of this lab. Health Department Bihar, who supported with the funding for our lab services to ongoing. Sri Krishna Medical College, this state-run medical college, within this premises, the lab is ongoing. Forbase Care are active collaborator. And pathology and medical oncology, all the medical oncologists who actually continued to send samples despite our very delayed reporting sometimes. So this was indeed a journey, very different journey that started with only 8 samples in a month in the first year. Sometimes the reporting time of around 2 months or more. But now we are able to process 30 samples in a month with the reporting time of 2 to maximum of 3 weeks. So this is indeed a journey, a very different type of journey. And this lab can be considered as a model that how a state-of-the-art diagnostic service can be provided in the remote area, in the rural area, to provide this service affordable by the patient. Thank you.
Thank you, Moitri, for this illustrated journey of your lab. But here I would like to mention over here that which I could not do as a macro dissection in Tata Hospital, which I started at Muzaffarpur, and actually yes, that improved the quality of
yes,
your...
Yes,
yeah,
correct.
so,
Yes,
and thanks to histopathologist who really helped us and supported us to get this macro dissection.
Yes, correct. Yeah.
Itself is so challenging and it is so time consuming, right? And you also mentioned the restriction of manpower and all. So very happy to say that at Genomic here we are working on developing an application with molecular scientists, computer scientists, NLP experts that brings together the NGS results with the latest guidelines, your annotations and everything, where we can interact in real time. So so that the reports are not stagnant. And
Yes,
at the same
correct.
time, it can bring down the time and the cost that's associated with the report. So
Yes, yes, yes.
I really hope that some places like these and everywhere, such kind of applications are really helpful.
Yes, I must say that this is possible just because of the handholding between among all the different kind of experts. I must say, pathologists, what you say, the analysts of other lab collaboration, developing the collaboration. So yes, everything is possible by the support.
Can you go
Yeah.
with that? Yeah, anything?
I think so. The importance of this presentation is not the part we are discussing. Importance is, anyone has been to Muzaffarpur? Okay. Oh, good. And where do you work? IIT Bombay. You have been to Muzaffarpur? Excellent. So you can relate to it. You don't have anything like this there. It is around 3 hours from Patna, and now the roads are good. Not the time when she went there. She's from Calcutta. Whatever has been made there, it was all by donation. Everything was donated by someone. Whatever you say, the structure made, the machine made, the premises. It is a Tata Memorial Hospital now, part of it. It was in the premises of a medical college, which is there. Other part, the testing, till recently has been almost completely free. Again, Moitri and the team got someone to donate money to see that the testing is free, for the patient. And I'll tell you the best part. And this is a bit of irony for being from Tata Hospital. The NGS lab started before the CBC lab started. So again, idea was this presentation gives you some glimpse, but what happened, that is the best part of it, that in that kind of a tier 3 city, if you have people willing to work hard, getting people, they can have it. So whole lot of kudos to Moitri and the team. Thank you so much.
Yes, I really admire Moitri because I was there 3 weeks when we started the NGS, right from installation to getting the service into the service for that NGS testing.